All right, we ended last time talking about the different systems in the plant, the shoot system and the root system, and I ended with the leaf system. I went through all the different morphologies and modifications to those different systems. And what I want to do now is take a step back and talk about sort of the processes that are happening in the plant, and specifically with plant growth, and how plant growth extends the plant body throughout its life. So just as a reminder, remember that plants are indeterminate. They grow throughout their lifespan, their lifetimes. They never stop growing. And plant growth in a plant is the result of two processes. The first of those processes is cell division. And so when I'm talking about cell division in a plant, these are repeated mitotic divisions, mitosis, that happen in a meristem. All those cell divisions are in a particular part of the plant called the meristem, and we'll talk about the different types of meristems here in just a minute. And just as a reminder, remember a meristem is a region of undifferentiated cells. This is the only place in the plant where active cell division is happening, is in the meristem, where we have this population of undifferentiated cells. The second process that's responsible for plant growth is actually cell expansion. So what cell expansion is this is growth, expansion of the cell, specifically with respect to the volume of the cell. So expansion of the volume of the cells. And this results in elongation of that cell and enlargement. So those two processes, cells dividing, followed by cell expansion, the volume of those cells actually getting bigger, so getting enlarged and elongated, are really what constitutes growth when we're talking about growth in a plant. So primary growth, remember, is that growth in length in the plant. So a plant getting taller or roots getting longer. And so in both the roots and in the shoot, the primary growth is happening in the apical meristem. And so in the plant shoot, we've seen this already. Here's the apical meristem. That's that population of cells that's dividing at that tip of the plant. In the root, which we can see on the right hand, the apical meristem is similar, where we have a population of cells that is continually dividing. And so we know that we talked about the three different types of tissues that we find in plants. We have the dermal tissue system, we have the vascular tissue system, and we have the ground tissue system that make up the body of the plant. Each of those actually have a primary meristem associated with them. And so within the plant, we have three primary meristems. And what these are partially differentiated cells that give rise to those three tissue types. So our primary meristems are partially differentiated. And give rise to those three tissue types. So just as a reminder, those are the dermal tissue, the vascular tissue, and the ground tissue. We went through earlier the main functions of each of these different tissue systems. So these three primary meristems are just below, just behind or below the apical meristem that we see in the plant. So remember that, the apical meristem giving rise to these three different tissue types And those primary meristems are located just a little bit behind the apical meristem. So the first of those is the procambium. And the procambium is responsible for giving rise to, is the primary meristem for the vascular tissue. And so as such, the procambium is a group of cells that is just behind the apical meristem. And when I say behind, I mean below that apical meristem. So these cells are actively dividing in the apical meristem. They migrate down, down the meristem, down that meristem a little bit, and then they're into a region where we find the primary meristems. And if those cells are partially differentiated into vascular tissue, they become that procambium, which will continue to divide and give rise to the vascular tissue. And so we see the procambium giving rise to our vascular tissue, and that's these lines that we see inside of the, where the vascular tissue is in the center of this leaf primordium. We see that vascular tissue that was given rise to by that procambium, that primary meristem. Just like we have with the vascular tissue, there's the primary meristem for the dermal tissue is called the protoderm. So this is for dermal tissue. And we see that that's just behind, but located toward the outside. And so that's giving rise to the dermal tissue that's on the outside of the, which is that outer layer on the outside of the plant. And this is happening in both the root and in the, both the root on the right-hand side here and in the shoot on the left-hand side. Now our last meristem doesn't actually have a fancy name. This is just the ground meristem, and that gives rise to ground tissue, as you might expect. And so the main point that I want to make here is that when meristematic cells divide, some of those meristematic cells stay in the meristem to maintain that population of cells in the apical meristem. Others of those migrate down and begin to differentiate. And differentiating is a developmental process that's used to create that specialized cell. So it's expressing certain genes to produce that distinctive structure and function. So in the apical meristem, we can maintain a population of cells that's actively dividing. As those migrate and begin to partially differentiate, depending on where they are and what genes are expressed, some will differentiate into that procambium, the primary vascular tissue meristem. Some will differentiate into the protoderm, that primary dermal tissue meristem, and some will differentiate into the ground meristem, or that primary meristem for the ground tissue. And so all three of these meristems then, of course, result in the, that primary plant body being comprised of the ground tissue, the dermal tissue, and the vascular tissue, each of them with their particular functions that we've talked about before throughout all of the plant parts, the shoot system, the root system, and the leaf system. Okay, so let's look at the organization of these systems the organization of each of these systems. And so when we look at our primary root system, this is organized in a pretty particular way. So as this root is growing to access more water and nutrients, we've got to be continually growing into new regions of the soil. And so we can think about this as the root pushing its way through the soil. And the root first does that, pushing its way through the soil with a group of cells that's on the outside of the apical meristem, just out to the tip, and that's called the root cap. And I didn't actually show you this, but in that microscope cross-section right here, we can actually see the root cap is this portion of the tip of the root. On the outside of that is the root cap. And the root cap has a couple of very specific functions associated with that. The root cap is providing both protection So as as the root is physically pushing through the soil, we actually need to protect it physically from protect the apical meristem from getting damaged as this is pushing through the soil. The other function that goes along with this is the lubrication. So the root cap will actually excrete some lubrication to allow this process to happen, allow the root to move through the soil a little bit more easily. The other part of the root system, root cap that's important is the root cap also is there to, also helps to sense gravity. And so there are some specialized organelles in the root cap cells that actually sense gravity and let the plant know which direction is down, which direction is up. That's our root cap. We see the right behind the root cap then is the apical meristem. And so the apical meristem is that part of the plant where all of that active cell division is happening. And so when we think about the root, the organization of the primary root system, we can, after the root cap, we can kind of organize this into these three different regions or zones of the root. And so right near the apical meristem, this is the zone of cell division. And so this zone of cell division includes the apical meristem, where all those actively dividing undifferentiated cells are, as well as those three primary meristems. So the three primary meristems being the protoderm creating the dermal tissue, the procambium creating the vascular tissue, and the ground meristem creating the ground tissue. So that's our zone of cellular division, including all of the meristematic regions. So that's all those cells dividing. The next zone just behind that, or just above that in the organization of the root, is the zone of elongation. So if you think back, remember I just said that plant growth is really two processes: cell division and cell elongation. So these are happening in both of in these two zones that I've told you sort of are responsible for all of that plant growth. So for zone of elongation, this is the zone that's responsible for pushing this root through the soil. As that zone of elongation. And so the cells here are taking up water, increasing them in length and expanding, and that provides the force that's used to push that root cap through the soil. So in that zone of elongation, you actually get that cell elongation, cell expansion, which pushes the meristem down through the soil. The last zone is this upper region here, when this is the zone of maturation. Sometimes you'll see this referred to as the zone of differentiation. And as you might expect, this is all the cells that are differentiating into the dermal tissue, the vascular tissue, the ground tissue. And so this is where final differentiation occurs. Into dermal, vascular, or ground tissue. And so because of that, this is actually the most important region for water and nutrient uptake. Which is one of the main functions of that root system. This zone of cellular maturation or the zone of differentiation is where all of the water and nutrient absorption in the plant is actually happening. And most of that is actually happening through the production of a special cell type that's a dermal cell that is produced in this zone of maturation, which are called root hairs. And so what root hairs are is these are epidermal cells. Sorry, I'll erase that. That are responsible for all of they're actually the point of uptake of all those water and nutrients. For H2O and nutrient uptake. So all of the water and nutrients that enter a root actually enter through the root hairs. Now, these are extremely important. And as you might guess, this importance is all in this increased surface area. So we take a quick look at what these root hairs look like and how those increase the surface area. All of these projections coming off of this germinating bean seedling here. These are all of the root hairs associated in that zone of maturation. And so you can see the root cap here. We can see the zone of cellular division and the zone of elongation, and then maturation starts right here, and we get the root hairs being produced. And how much that increases the surface area is really quite impressive. Okay, so that's the organization of our the primary root system from the outside. Let's now take a look at the cell types and the tissue types that we see in a cross section. And so here's a cross section of two different roots. On the left-hand side, we see a eudicot root, and on the right-hand side, we see a monocot root. And if you remember back to a couple of lecture videos ago, this difference, monocots and eudicots, these are two major branches in flowering plants. Just like we had with venation, we had parallel venation in the monocots, and we have reticulate venation in the dicots. There are organization of the vascular tissue in both the roots and stems that are different in monocots and dicots, and so we often talk about these two major groups and show the variation that we see in those. And so in a lot of these slides, when I'm looking at shoot systems and root systems, I'm going to be showing you what we see in monocots and what we see in dicots. So on the left here is eudicots, and on the right-hand side, we have monocots, but we can label those same parts throughout. And so if we look at the primary organization of these root systems, these are cross sections. So I'm taking a root, I'm cutting across it, looking down at it through a microscope, and I've stained this so certain cell types stain differently than other ones to try and differentiate which cells are which here. So if we start from the outside and head in, we see the epidermis on the outside. So this is the very outer layer of cells. There's our epidermal cells. This is one cell layer thick, as you can see, working its way all the way around that cross section of the root. The majority of the ground tissue that's in the plant is made up by, or in the root is made up by what we call the cortex. The cortex is part of the ground tissue that's in there. We also have, and so that's I'll point a couple of these out in some of the other cell types, but just to mention some of these cortical cells, you can see all these starch granules that are being stored. So that's energy storage, all those little purple dots in there in both the monocot side and in the dicot side. These are all starch granules being stored in that cortical tissue. As we move in, the next layer is the endodermis. The endodermis is a layer of cells that seals off the vascular tissue from the rest of from the cortex itself. When we talk about water transport, this becomes an important part of the anatomy of the root system. Just inside of the endodermis, there's a layer, and I'll show you this in the next slide a little bit better, called the pericycle. And so I'm just going to pop out here and just inside of the endodermis is the pericycle, and the pericycle is actually another type of meristem. And so this is only found in roots, but it's a meristem that is responsible for root branching. So it's responsible for initiating root branching. Now, like I said, I'll come back to this one on the next slide so you can see this one a little bit. This whole region in the center, inside of the endodermis, we call the vascular bundle or vascular cylinder. And so the vascular cylinder is the endodermis and everything inside of it, and that's the vascular tissue inside of the root, and it's always in the center of the root. In the center of the root. And when we talk about water transport, like I said, we'll talk about why this is there. And then we have in the vascular cylinder, we of course have both the xylem, responsible for transporting water, and the phloem, responsible for transporting our photosynthate and sugars. Now, in a monocot stem, we have this region of the monocot stem in the center of the vascular cylinder, which is another part of the ground tissue, and that's called the pith. And so in monocots, you have a separate set of ground tissue right in the center that's called the pith. Okay, so I'm going to zoom in on a couple of these just so you can see these a little bit better, but I'll just emphasize a few things here. One is that in the roots of angiosperms, both monocots and dicots, we have the vascular cylinder in the center of the roots. The main difference between monocots and dicots is that if we look at a monocot stem, monocot root cross-section, there's that vascular cylinder in the center. The xylem forms this sort of X shape, and those are those really big, thick, walled, dark staining cells. Those are all the xylem. The phloem, on the other hand, is in between each of these xylem arms, and it's this blue-green, these blue-green cells that don't stain as darkly. Those are our phloem cells moving that photosynthate. If we look at the monocot root system, we actually see the xylem are these large cells, these large openings here. Those are our xylem tubes that are moving water throughout the plant. And then the phloem is actually spread around the outside of this vascular cylinder. We see the phloem. So our xylem is arranged in a ring, these large xylem elements, and then interdigitated with the phloem toward the outside. But in the very center, we have just ground tissue. That is actually the pith part of that cross section. And so, just looking at this a little bit closer, we can see the pericycle, where if we zoom in on here, I'll zoom in on this bottom corner down here. We see, zooming in on that vascular cylinder, we see the endodermis surrounding this, and then just inside of the endodermis is a layer of cells. That's that meristem responsible for initiating root branching. That's the pericycle itself. And then here we see the xylem and the phloem. Now, the reason I want to show this is that for a lateral root to emerge out of a root, we actually have to have the vascular tissue be connected. And that vascular tissue in the root is running down the center of the root. And so because it's running down the center of the root, you actually have to initiate the vascular tissue in a branch root. You have to initiate from that vascular cylinder. That's why the pericycle is there, initiating that root growth. And so here we see an emerging lateral root that has just popped out of the pericycle and is emerging its way through the cortex. As that grows, you'll actually see that lateral root being, busting its way through the cortex and will branch out from the root. That's important because now this vascular tissue that's running through this lateral root, the root's being cut in a longitudinal section, is connected to the vascular tissue of the root that it branched from. And that's an important connection to be able to have our vascular tissue all interconnected throughout the whole plant. That's why that pericycle is an important one. Okay, so let's move now to the shoot system and look at the primary shoot system. This is a reminder slide here showing these newly forming leaves, or these leaf primordia. And the apical meristem at the shoot, and then the apical meristem in these lateral buds, as well as our primary meristems, those partially differentiated procambium, protoderm, and ground meristems, creating these dermal, ground, and vascular tissue systems. And so just like we did with our roots, let's look inside of these and let's look at a cross-section of a stem. And so here's a cross-section of a eudicot stem, and showing many of the same parts that we saw in the root. And so on the outside of this stem cross-section, we have the epidermis. Each of these, arranged in a ring around the stem, are vascular bundles. So those are the vascular tissue. And we know that our vascular tissue is made up of xylem cells toward the inside, and then phloem cells, the blue stained ones here, toward the outside of that stem, and that running through in between them would be the vascular cambium. Associated with each of these vascular bundles, you see this group of red cells that are out at the out towards the outer end. These are actually called fiber caps, and they are oftentimes made up of cells that are a little thicker walled, which is why they stain darker, and they provide some structural support to that growing plant. We see the xylem and the phloem and the fiber caps in those vascular bundles separated by that vascular cambium. The rest of the plant then is made up of ground tissue, and that ground tissue is separated just like in the root, is separated into the cortex outside of the vascular bundles, outside of that vascular tissue, and then toward the center of the stem we have the pith. So cortex and pith, just like in the root, make up the ground tissue in the stem. Okay. So that sort of rounds out the primary root system and primary shoot system, going through all of our different cell types. In lab, we would have been making tissue sections and making cross sections of these, staining them with different stains, and looking at all of these different cell types. And so something that I do expect you to be able to do is to look at a cross section of a stem like this, or a cross section of a root like these, and be able to label the different parts: epidermis, cortex, endodermis, the vascular cylinder, xylem, and phloem. Be able to identify and label these cell types throughout the plant, both in the roots and in the shoots. And then being able to recognize the differences between monocots and dicots are important. And so here I've shown you the cross section of a dicot. What would this look like if it was a monocot? And I'll just go ahead and draw in part of the monocot. What was one of the main differences with the vascular bundles in the dicot versus the monocot? Well, in dicots, you remember, these are arranged in a ring like we see here. But in monocots, they're not arranged in a ring, and the vascular bundles are actually scattered throughout the plant. So we would still have our epidermis on the outside. The vascular bundles are still divided between those dark staining, thick-walled xylem toward the outside and phloem toward the inside of the stem. And each of these is a vascular bundle. But rather than being arranged in a ring, the vascular bundles are scattered throughout the stem. So just for clarity for later, this is what a monocot would look like. And then, so what's the rest of the tissue that we see in here? Well, the rest of our tissue type that we see in here is all cortex. So whether it's in the center or not, since we don't have a defined ring where we see outside of that the cortex and inside of that the pith, all of this tissue throughout is cortex tissue in that monocot stem. Okay, I'm going to stop this one here, and then we're going to move into looking at plant cells and the tissues that make up the primary plant body for the next